How was the exam? Better than what I thought? Okay.
You tell me.
Well, I don't even know at this point. They're still being graded. So that's why I'm kind of curious to hear your perspective. What did you think about it? I see a lot of people shaking their head yes. That's always a good sign. That means like, you're an idiot, Ahern. Yeah. Comment? I'm sorry. A couple throw you off a little bit. Okay. Okay. I don't take things personally, so if you have things you like or don't like, I always like to hear them. Any comments?
Clear.
Clear? Fair? Okay. Unfair? Okay. As I said, I don't take things personally. I think that one of the reasons I ask this, I always ask after an exam, is that I learn a lot about giving exams from year to year. And one of the things that you always hope for in writing an exam is that it's clear, excuse me, and that students understand what you're asking, and that it's fair. I think that's two things that are really important. And I will tell you that having written many, many exams over my career, there are no perfect exams. Much as you would like to have a perfect exam that is absolutely great at assessing ability, there are no perfect exams. Exams can be good, and exams can do a pretty good job, and I hope my exams do a good job of that. But there are no perfect exams. So I think it's important as instructors that we listen to you a little bit. Not even a little bit, a lot, okay? That we listen to you and understand what you don't understand, and understand where maybe we can make exams better. Because it's important that exams be fairly testing people's ability. And I think that's a very important consideration. So your feedback, even though you're not giving me any, is important. Yes, sir?
I guess the criticism I had was I didn't like the question about the duties that have to do with the person holding it properly.
Okay.
I felt like it was testing our knowledge on the subject matter more. It was more specific to the disease.
Okay, so didn't like the question on the protein folding problem. Okay. All right. Yeah. More time, yeah. How many people felt time was a factor? Okay, yeah. This exam, it tends to happen with that because people tend to take a little bit too long in the calculation, and I think that's a factor. It's usually not a factor on the second exam. It's never a factor on the final. And the good news is that next term, it's a completely different format of exam, and it's never a factor there either. So this one exam that you've had is the place where it does tend to be a factor. We tried a few years ago to get an evening exam with this, but evening exams are still only supposed to go for 50 minutes, so it doesn't buy us out of the problem that we have of the length. So it's an issue that I've wrestled with a little bit, and I don't make it long for the sake of long, but here's what I always ask students. Well, if I say, would you like to have it shorter, almost everybody would say yes. And I said, okay, then that means each question's worth more points. Do you want that? And almost everybody goes, no. So it's a tough call. And so writing that means that you have to find that right balance. I originally wrote the very last question to have 3 questions instead of 2. And when I looked at the exam, I thought that's probably a little bit too much. And based on what I'm seeing here, that probably would have been a little bit too much. So I'm glad I left it out. So that was good. Yeah? How do I do the curve? Well, I look at where the distribution of grades are, and I just decide based on that distribution of grades that people, on the first exam, it's not quite so clear, but as we go further along, we'll see groups of things that will happen. And so I try to assign things on the basis of groups that I see there. I don't have any fixed numbers of A's, B's, C's, D's, or F's that I give. And I do try to make it as best as I can with those groups. So, and that's as good of an answer as I can give you, because that's really literally what I do. I do look at the overall average in considering where those breaks might happen and so forth. And so students always say, well, how can I improve my grade? And that'll happen obviously after this exam. The answer to that is always by improving against the average. So if you improve your score against the average, then you're going to move up. All right, so if the average, as I tell students, well, if the average on an exam is 10 and you make a 90, well, you just really moved yourself up a lot, okay? And students will always say, well, I want an easy exam. Well, in fact, the best exam for you to move up on is a hard exam, because the lower the average and the better you do against that low average, the better off you will be. Does that make sense? So, and no, I don't make them hard. One of the most difficult things I have in writing an exam is I can never predict what the average is going to be. I've tried, and I have tried, and I've tried. And I've written exams that, oh, that's a real easy exam, you know, and then you'll see the scores, and it'll be down in the toilet, you know. Or you'll write an exam and think, well, that's going to challenge them a little bit, and then you'll see the highest score ever. So, I mean, I'm the worst person at guessing those, so I just write what I think are appropriate questions. I don't try to steer it one way or the other, because I can't do it. I've tried. So, does that answer your question? Okay. If you have feedback, feel free to send me an email or whatever. I'm always happy, as I said, to take that, and it does help me, I think, to write better exams. So I like to hear that. All right. Well, we are well ahead of where we need to be, and I like to stay that way. So being ahead is better than being behind. So we'll finish the catalytic strategies today a little early, and we'll start the new material as well. But we have some things we have to finish in catalytic strategies. So let's go and do those first. The carbonic anhydrase I talked about last time is an example of an enzyme that uses nucleophilic attack to accomplish what it does, which is converting carbon dioxide into carbonic acid with the help of water. And we talked last time fairly quickly about this mechanism. So this mechanism that you see on the screen is what's occurring in... Hello?
Excuse me.
Oh, okay. Oh, she ducked this time. Okay, well. All right. So the mechanism is of what's happening here. We're looking at the active side of the enzyme. And in the active side of the enzyme, we have a zinc that is held in place by 3 histidines. The zinc is a positively charged ion, and that positively charged ion helps to hold water into place. Okay? So it's helping to hold water into place. That we can see that coordination right there. We remember, of course, in a water molecule that it's asymmetric, and that it's asymmetric for charge as well, meaning that the oxygens are partially negatively, the oxygen is partially negative charged, and it's attracted to the positive ion of the zinc. The most critical step in the process actually occurs in this very first step, which is the ionization of the water. And as I noted last time, the ionization of the water, that is the loss of that proton, occurs much more readily at a higher pH than it occurs at a lower pH. And it is for this reason that the enzyme works more rapidly at a pH of 9 than it does at a pH of 7. It still does very well at a pH of 7. You remember that the turnover number at pH 7 was still 600,000 per molecule of enzyme per second. Okay, so it's still working very rapidly, but it'll work even more rapidly at a pH of 9 because this first step happens. Well, the creation of this hydroxide ion in the active site is a necessary step for the reaction to occur because that hydroxide ion, like we saw before with the alkoxide ion and also with the hydroxide ion in the chymotrypsin, acts as a nucleophile attacking this carbon inside of this carbon dioxide. The attack of that carbon results in a bond, covalent bond between the carbon and the oxygen that you can see right here. And that is actually the step that's creating the bicarbonate that is released here. So the bicarbonate has been formed by this point, water comes in and releases the bicarbonate, and we're back right where we started. So that's what's happening in carbonic anhydrase. And as I said, it's a very rapid process that this goes through. Okay. Yeah, I don't think I want to go through that. And we'll leave that all be. Okay. So that's the basic process that's occurring in the catalytic action of a carbonic anhydrase. Okay. Well, I want to turn our attention now from carbonic anhydrase to yet another group of enzymes that are very interesting enzymes. I want to say a little bit about them in general before I talk about their mechanism. But we'll see that the mechanism that they use is what we've been seeing the themes of so far. We're going to see an activated water acting as a nucleophile attacking, in this case, a phosphodiester bond. Okay? So let me just first give you some general information about restriction enzymes. Restriction enzymes are enzymes that are found in prokaryotic organisms, that is bacteria. So bacteria contain restriction enzymes. Restriction enzymes are also called restriction endonucleases. And restriction endonucleases have a very, very interesting and useful property. Okay? The interesting and useful property is that they recognize specific sequences of nucleotides in DNA. They recognize specific sequences of nucleotides in DNA. They bind to those sequences, the specific sequences, and they cut at those specific sequences. Okay? So they recognize specific sequence, they bind to the specific sequence, and they cut at the specific sequence. Now you might wonder why they do this. DNA is pretty important, and the reason that they do this is because this is a defense mechanism for the bacteria. A defense mechanism for the bacteria. How does that work? Well, bacteria get infected by viruses just like we get infected by viruses. Many of you are infected with a cold virus right now. Okay? We have an immune system that goes out there, and though it takes a while to get operational and to actually do its job, the immune system eventually takes over and we get better because the immune system is protecting us from invading viruses for the most part. It's not a perfect system, but it's a pretty good system. We can get a vaccine, for example, and our immune system is what gives us the protection against infection again by that same virus or organism. All right. Well, bacteria are single-celled organisms. They're lone rangers, right? They don't have an immune system. They don't have the ability to have an immune system because an immune system takes a multicellular organism and it takes a tremendous amount of cellular energy to do that, working together, coordinating to make antibodies. Bacteria don't have that option. So every bacterium is for itself. They can't go out there and coordinate an effort against a virus or even another bacterium. All right? Well, what they have is, since it's every bacterium for itself, is that they have these restriction endonucleases. So if a virus comes along and tries to infect the bacterium that has a restriction endonuclease, restriction enzyme, okay, then this system kicks into place. Okay? So the virus inserts its DNA into the bacterium. That DNA goes into the bacterium, and if there's nothing present, the DNA of the virus will take over and start making copies of the virus and kill the cell. If the bacterium makes a restriction enzyme that recognizes a specific sequence in the DNA of the virus, then it will cut it. Well, cutting it means that you are interrupting the genetic information of the invading virus. The invading virus's DNA is no longer functional, and you have just stopped the virus in its tracks. Okay? That's a pretty cool system. Well, you might sit here and think, well, that's really great, but how in the world does it keep from cutting its own DNA? Because if the restriction enzyme is recognizing a specific sequence, won't it cut that same sequence in its own DNA? And the answer is, if there weren't other protection, it would. So, a very common restriction endonuclease is called HIND3, H-I-N-D, and then the Roman numeral 3, HIND3. Okay? HIND3 recognizes the sequence AAGCTT on one strand. Okay? You don't even know that sequence. It doesn't really matter. Okay? But that's what it recognizes. That is a 6-base sequence. And if we say that sequences occur in DNA relatively randomly, that sequence will occur on a random basis once every 4,096 nucleotides. So on average, that sequence is going to occur once in about every 4,000 nucleotides. Okay? If a virus, some viruses have a DNA sequence that might be 30,000 nucleotides long, 40,000 nucleotides long, even 150,000 nucleotides long. Okay? On average, it's going to have multiple of those HinD3 sequences within it. So if the bacterium has this enzyme and it can cut that sequence, then it is very effective at stopping that virus from doing its thing. So how in the world then does the cell protect itself from cutting up its own DNA and cutting only the viral DNA? Okay? Well, the bacterial cells have a very interesting protection for this. So when we talk about restriction endonucleases, we also have to talk about a protecting enzyme called a methylase, M-E-T-H-Y-L-A-S-E, a methylase. The whole system I'm going to describe to you is called restriction/modification. So restriction/modification. So I've told you what the restriction is, that it's recognizing and cutting specific sequences. What's the modification? The modification is made possible by the enzymes known as the methylases. A methylase recognizes the same sequence as the restriction enzyme, and instead of cutting it, it puts a methyl group in it. So somewhere in that sequence, it varies from enzyme to enzyme, but somewhere in that sequence, it puts a methyl group in there. Why is that important? Well, once the methyl group is in there, the enzyme can't cut that DNA if it has a methyl group in it. So the methylase methylates the host DNA. At every place, there's an AAGCTT. So the host DNA doesn't get cut by the restriction enzyme. The invading viral DNA, unless it gets methylated first, which can happen, unless it gets methylated first, its DNA is naked, it's unprotected, the restriction enzyme is going to cut it, and the virus is going to be stopped. Does that make sense? So it's the combination of the methylase and the restriction enzyme that give the cell protection. Yes? So the question is, does it insert the methyl group into the sequence or onto part of the sequence? It's actually modifying a base within the sequence. So it's putting it onto one of the nucleotides in the sequence. Yes?
In the event that the viral DNA does get methylated,
yeah.
Does that inactivate some part of its functionality?
A very good question. So her question is, if the viral DNA gets methylated, does that inactivate any part of its functionality? Methylation actually can have some effects on gene expression, but the biggest effect that would happen here is that the restriction enzyme wouldn't work on it and the virus would actually kill the cell. So if the methylase wins the race, if the methylase gets to the viral DNA before the restriction enzyme gets there, that cell is toast. Okay? Because that viral DNA is now going to be protected, it's going to make copies, it's going to basically kill the cell. Okay? Yeah, where you at? Down here, okay. Yes. Okay, good question also. Once the viral DNA gets methylated and starts copying itself, are the copies all methylated? Okay. The answer is probably not. All right. So there will be some protection that's there because perhaps as new copies are being made, that new enzyme is going to have a shot at getting at them. So it's a complicated system and it's not a perfect system. I find that we always tend to want to think about a perfect system, right? That is, that the methylase never wins the race, and the restriction enzyme always does the cutting, but we don't have that. We don't have that with our own immune system. We've seen HIV as a prime example. We don't have perfect protection, but we have a lot of protection. And the bacterial cells that have restriction enzymes with a modification system, in fact, have an awful lot of protection as well. Yes? Another good question. Are the restriction enzymes and the methylations in the cell relatively equal? That'll vary a lot between restriction enzyme and modification enzymes. But for the most part, I think that the restriction enzyme is probably a little bit more abundant. Yep. Yes?
So what determines which reaches the virus first, like methylation?
What determines which one reaches the DNA first? You're going to like this. The process of diffusion. So it's a chance. It's a chance thing. So again, when we think about this, in the overall scheme of things, an individual bacterium may die because it doesn't win the race, or doesn't have the right one get there first. Okay? But overall, you're going to have bacteria that will survive that otherwise wouldn't survive, and that's why the system persists. Does that make sense? That's another hand. Yes?
Is there a high enough concentration of enzymes in the bacteria that the virus could not kill the bacteria even if the enzyme present?
So his question is it possible for the virus to kill the bacterium even though the enzyme is present? Yes. Okay. So there's a variety of scenarios that we could imagine will happen with this. And as I said, it's not a perfect system, but it does ensure a greater likelihood that bacteria that have this will survive. Yep. Okay, good questions. Now, remember what I said when I started this. This is a system present in bacteria. We don't have this in our cells. We don't need this in our cells. We have an immune system for that purpose. Okay? So eukaryotic organisms do not have restriction modification systems. Only bacteria have restriction modification systems. Okay. Well, now that I've given you some background on what it does, let's look and see how it does it. Okay. All right. So we're going to focus on one enzyme called EcoR5. I've talked about Hindi3. Here's another one, EcoR5. I don't like this figure. Well, I'll leave it there for the moment. Okay. EcoR5 is an enzyme that recognizes a sequence. We don't even need to know the sequence at this point. But if there's a methyl group in the middle of this sequence that EcoR5 recognizes, EcoR5 will not cut. And there's a reason why it doesn't work and why it doesn't cut. And it relates to the mechanism of action of the enzyme. Okay? We're going to see that in the action of this enzyme, there's two things that play very important roles. The ion magnesium is one, and the other is the molecule water. And like what we saw with zinc, where we saw zinc activating water, holding water in place, and then having ionization and the ion attacking the carbonyl carbon of the carbon dioxide, all right, so too does the magnesium help to hold water in place so the same ionization can happen and so the same nucleophilic attack can happen, only this case it's on a phosphodiester bond. This is simply showing the magnesium ion that plays a role in this, and it's a kind of a higgledy-piggledy figure. But magnesium is playing an important role in ultimately helping to hold water. Okay? Now, this actually shows the sequence that's recognized. As I said, you don't need to know the sequence, but the sequence that this enzyme recognizes is GAT-ATC. And most restriction enzymes, by the way, recognize a symmetric sequence. And when we say symmetric, if we read it 5' to 3' on the top strand, it reads the same way 5' to 3' on the bottom strand. Okay? Molecular biologists call that a palindrome. It's technically not a palindrome because radar is a palindrome because it reads the same way backwards and forwards. But this reads the same way backwards and forwards if you read one strand and then you read the other strand. Okay. The methylation that's happening in this enzyme is happening right in the middle of this sequence right here. Okay. One of the things that happens during the catalytic action of an enzyme is there's a couple of steps. The enzyme actually finds its sequence on a DNA in an interesting fashion. It grabs a hold of any old part of the DNA. As the restriction enzyme grabs a hold of any old part of a DNA, and then it starts sliding down the DNA sequence. It's sliding down that DNA sequence. And when it finally hits the sequence that it recognizes, it stops. Okay? That's kind of a cool way of finding the thing that it's going to cut at. When it gets to that sequence that it recognizes and it stops, it not only stops, but it undergoes a conformational change. Because why? The proper substrate has bound to the enzyme, just like you saw with the serine proteases. Okay? The proper substrate has bound, in this case, the sequence GAT-ATC. The binding of that proper sequence in the active site of the enzyme causes the enzyme to undergo a conformational change. And the conformational change causes the DNA to bend right in the middle of where it bound. Okay? Right square in the middle. Everybody with me? So it's bound to the DNA, it's slid to its proper sequence, it's bound to the proper sequence, it's stopped, it changed its shape, and now it's holding on to this bent DNA in the middle of it. Okay? That's what this guy's doing. Now, this bending turns out to be critical for the cutting. The restriction enzyme, remember, is going to cut. We're talking about the restriction enzyme here. We're not talking about the methylase. We're talking about the cutting by the restriction enzyme. It's bent this. All right, if we were to look at this bend inside of this DNA molecule, what we would see is that there's a nice little position about right here where my pointer is, underneath the middle of this thing, where a magnesium ion will be held in place. And right above that magnesium ion, there's a water ion that will be held in place. And this will only be created, this little pocket will only be created if the DNA is bent, and the DNA only bends if the proper sequence is bound by the enzyme. Well, the water, like we saw before with the zinc in the carbonic anhydrase, the water gets ionized, magnesium is holding it in place, the ion attacks the phosphodiester bond and causes it to fall apart. All right. Because it has done that, we've broken a phosphodiester bond, and phosphodiester bonds are what hold DNA together. Now, what you've done is you've broken the bond, the strands come apart. You've just cut DNA. So that's how the cutting actually occurs. That's the mechanism of the cutting. I'll talk about the methylase in just a second, but the cutting occurs by that mechanism. Questions about the cutting? Yes? Very good question. There are two strands here. Does this happen twice? And the answer is yes, it does, and yes, it must. We have to cut both strands to completely cut a DNA. Yes? Is it possible for ligase to come in after this? Ligase is an enzyme that will join phosphodiester bonds. It is possible for that to happen, yes. Kimberly? The enzyme has no effect on the rest of the DNA. It's only going to have an effect at the place where it binds the proper sequence and causes that bend to occur, because that bend is necessary for the action that we're talking about here. Yes, back there. Okay, we're getting into things we'll talk about next term. But his question is, does a topoisomerase, which is an enzyme that unwinds and untwists DNA, does it work in a similar fashion to this involving a bend? The answer is no, it does not. Yes? Yeah. Okay. So she's asked a question here about there's different enzymes that recognize things in different ways. Let me go back to the sequence and show you what she's asking about here first. Okay, so if we go back to the sequence this guy recognizes is GAT-ATC. All right? And this particular enzyme cuts between the T and the A and between the A and the T. All right? So it makes a perfectly symmetrical cut in the middle of a perfectly symmetrical sequence, which results in what we call blunt ends. Okay? The ends are exactly the same sticking out here. Some enzymes, some restriction enzymes don't do that. Some restriction enzymes will cut between the G and the A here and the G and A over here. Her question, I think, was why that happens. Was that the question? Yeah. Okay. The answer is that's just simply the way that they evolved. So it has to do with the way that the other strand gets cut. Okay? And so some enzymes, if they cut here and they cut here, because they're both cutting between a G and an A, leave this end has an ATAT hanging over, and this has an ATAT hanging over, but they're not blunt like we had before. So don't want to get too hung up on the recognition sites themselves, but suffice it to say that enzymes have different places within a sequence that they will cut, and the implications of that are what the overhangs look like. We're not going to get too much more into that. Yes? This is all still bacteria. Everything's bacteria. Okay. Other questions? Let's go back then and talk about the effect of methylation. Okay? So here's what happens in the case of the methylase that protects EcoR5 sites. All right? It modifies the adenine right there. Okay? So it's putting a methyl group on the adenine within that sequence, as you can see. And that methylation of the adenine stops the enzyme from working. And what the methylation is actually doing is getting in the way of this coordination of the magnesium and the water. So this very important structure that we needed to have for cutting is not stable when the methyl group is present there. The methyl group interferes with it. So it's a very effective way of stopping an enzyme from cutting. And by stopping the enzyme from cutting, the cell's DNA is protected. I see a little bit of nodding. That's good. Hope you're not nodding off. Right? The last step before you fall asleep, right? Okay, other questions about that? It's a cool system. Yeah? What happens at enzyme? It's a very good question. What happens at enzyme when it encounters a methyl group? It won't bend. It doesn't recognize it as a proper sequence, so you don't get bending, you don't get any of the things that we saw here. So EcoR5 goes along, and it's just as if it didn't see it in the first place. Very good. Okay. Well, those are restriction enzymes. Let's see. Maybe we should sing about that, I think. I have a song about this. We have never sung this song in class, so it's a relatively new song. It's the first time you guys will be the first people ever to sing this song in class. So it's about what we just heard about, and it's about the enzyme Hindi 3. It's to the tune of an old song called Chim Chim Cheree. Everybody know that song? Okay, here we go. I'm obsessed with I can't start. Let me think about it. I'm obsessed. Now I can't get the tune in my head. Somebody want to get me started? I'm obsessed with AAGCTT because it's the binding site of Hindi 3, cutting up DNA most readily. The buns are not blunt when they're cut up, you see. That's the wrong song! Okay. I'm obsessed with AAGCTT because it's the binding site of Hindi 3, cutting up DNA most readily. The ends are not blunt when they're cut up, you see. 5' overhangs of AGCTC. This is awful. Bacteria don't have an immune system, so they must fight off phages or they will not grow. Protection of copying is their strategy. And one of their cutters we call Hindi 3. On binding of AAGCTT, the recognition sites bent easily. Phosphodiester attacking, meanwhile, has water behaving as nucleophile to stave off the phage for a little while. Well, these enzymes cut cells' DNAs. The answer's provided by a methylase, adding a methyl group on top of what? The sequence these enzymes would otherwise cut. So cells get protected in this simple way from nuclease chewing of their DNA. The phage is not lucky in most every case unless methylases win the enzyme race. If that happens, then the cell gets erased. All right. Yeah. That was bad. You can tell I've never sung that song before. God, that was awful. Okay. You know, the worst thing is that you videotape this and it'll be on YouTube forever, you know. Yeah. Oh, well. I've done stupider things, I think, on YouTube. All right. So the last thing I'm just going to mention briefly, I'm not going to go through any mechanism because I think it's really more than what we need to be going through and doing. But they're just a group of proteins that are very important in what they do. And I'm just going to say a couple brief words about them. Myosins are proteins that are involved in translating ATP energy into movement. That's what they're there for. I'm moving around up here because of myosins that are translating ATP energy into movement. My legs, muscles are contracting thanks to myosin. And that's a very, very important consideration for, obviously, for animals. Okay? And we see myosins occurring throughout all of biology. Even single-celled organisms like slime molds, for example, have myosin within them. I'm not going to say anything about them because I later will talk, and it'll actually be next term, but I'll talk about enzymes called ATP binding enzymes that have specific structures that enable them to work. These enzymes have what are called P-loops, and they're very specific structures for holding and manipulating ATP. So we're going to save that until next time. So what I'm going to do at this point then is move forward and talk about catalytic strategies. So we're getting deeper and deeper, as you can see, with the reactions that we've been talking about. We first talked about general things about protein structure. Then we talked about mechanisms by which enzymes have acted. That's been the past couple of lectures. And now we're going to turn our attention to controlling enzymes. Because as I've alluded to earlier, enzymes are able to catalyze things so rapidly that cells need to make sure that they don't let too much product or too much substrate be used up. So they have to control those. And so they have some very interesting and elaborate schemes that we'll see for that regulation. If you want to understand how cells do what they do, this is going to be the most important thing. The control of enzymes, whether it's the way enzymes work after they're made or the determination about whether or not they are made, really are how cells function. Okay? This is where it's all at. So this term we talk about the ways in which the enzyme is made after it's been synthesized. Next term we'll talk about the ways in which the enzyme synthesis itself is controlled. Okay? That's called gene expression, and we'll talk about how that's done. So for right now, we're going to talk about how the enzymes are controlled once they've been made. Okay. Well, a couple lectures ago, I gave you a term that I'm going to give you in a lot more detail here. It was called allosterism. Okay. Anybody remember what allosterism meant? Anybody memorize that definition for the exam? It's one of my most common exam questions. I didn't ask you, but I almost did. Yes. Was that you? Oh, you're not doing it. Okay. Yeah. Okay. So they are allosterism itself. You're on the right track. They have enzymes have binding sites on them for molecules. We can think about these occurring in two ways. One, we can think about binding sites for the molecules the enzyme acts on. And the other is we can think about binding sites on the enzyme for molecules that bind that affect the enzyme, that it doesn't act on. Okay? So allosterism is actually the latter. That is, that allosterism is, I'm going to give you a definition again because I think it's a very important definition. All right? Allosterism is a process whereby a small molecule binds to an enzyme and affects its activity. Process whereby a small molecule binds to an enzyme and affects its activity. That effect can be positive. That effect can be negative. Yes, question? What's the difference between cooperativity and allosterism? Good question. Cooperativity is what we refer to in the case of hemoglobin, where it refers simply to the binding of what the protein binds to. Right? They're different colors of the same thing. Okay? But we'll see allosterism can actually work with molecules besides what the enzyme binds to. Okay? That's what allosterism, how it really differs. Okay? And we'll see that in just a minute. Okay. So allosterism is pretty critical. Now, as I go through this, I'm going to say some things that are going to be inaccurate. All right? You like this? But before I get started, I'm going to tell you something that's inaccurate, something that's wrong. Okay? That inaccuracy is that this mechanism I'm talking about, allosterism, is not an on-off switch. But I'll talk about it like it is. I try not to do that. But I want you to remember, when we talk about allosterism controlling an enzyme, we think about it in simple terms. The enzyme is turned off or the enzyme is turned on. All right? It's much more like a radio whose volume is turned loud or whose volume is turned soft. Okay? So it's more a matter of degree when we talk about allosterism. Yes? So his question is, are the small molecules that bind to enzymes that cause the enzyme to be more active known as coenzymes? And the answer is no, they're not. Coenzymes are separate things. Okay? Okay. All right. So the classic enzyme we use to describe allosterism is called ATCase. We'll talk a lot more about it next term, but I'm going to introduce it to you this term. Okay? ATCase, I'll give you the long name for it, which you won't need to know. But ATCase stands for aspartate transcarbamylase. Okay? For what that's worth. Yeah, don't you like that? All right. What does it do? That's the most important thing to start with. Well, aspartate transcarbamylase, or ATCase, I'll call it ATCase because it makes you laugh if I say the long one. All right. ATCase combines two small molecules and together to make a bigger molecule. One of those molecules is aspartate, the enzyme, the amino acid, aspartate. That's where the enzyme, the A part of the enzyme gets its name. Okay? There's aspartate. Aspartate, I'll take it back. Aspartate is, now I'm going to embarrass myself and see which one it is. It's this guy here. It's this guy here. Okay. Anyway, aspartate is one of the substrates for the enzyme. All right? The enzyme takes these two guys, puts them together, and makes a bigger molecule. All right? That's what ATCase is doing. Well, why is this important? Well, this enzyme is catalyzing the first reaction in a pathway that leads to the synthesis of the nucleotide CTP. Now, notice what I just said. I said it catalyzes the first reaction in a pathway that leads to the synthesis of CTP. It is only the first reaction. And notice, it does not catalyze the synthesis of CTP. CTP happens only after several more steps. So if I ask you on an exam, does ATCase synthesize CTP, I hope you will tell me, no, it does not. Because it does not. It synthesizes a molecule that later, after many steps, can be converted into CTP, but it does not catalyze the synthesis of CTP. Now, why am I being picky about that? Well, I'm being picky about that because you're learning for the very first time what a metabolic pathway is like. When we talk about a metabolic pathway, we talk about a series of steps that lead to a final product. All the things we've talked about so far have been this goes to that. We saw a serine protease took a protein and it cut it in half. We saw a restriction enzyme took a DNA and it cut it in half. We saw carbonic anhydrase took water and CO2 and put them together. Each of those was one step. Okay? A metabolic pathway would take the product of one step and use it as the substrate for the next step. And then the next step. And then the next step. Right? So the product of one reaction becomes the substrate for the next reaction, which creates a new product, which now is the new substrate for the next one, et cetera, et cetera, et cetera. All right. Well, that turns out to be very, very convenient and controllable. Very convenient and controllable. How so? Well, it turns out that the end product of this pathway is the nucleotide CTP. This is the last thing that's made at the end of this pathway. Cells are very careful in how much of any given nucleotide they make. We'll see this next term. I'll tell you next term repeatedly that cells have greater controls on how much of each nucleotide they make than anything else they have inside of them. Why do they care about how much of any given nucleotide they make? The answer is that if they make too much of a given nucleotide, they're much more prone to having mutations happen. They're much more prone to having mutations happen. So the best defense against mutation is keep everything balanced properly. Cells don't want to make too much CTP, just like they don't want to make too much GTP, and they don't want to make too much ATP. Making too much or too little of something is going to favor mutation. All right. Well, look at what this CTP does. You see this little red arrow? This little red arrow up here is to indicate that CTP binds to ATCase and turns it off, which means it turns its volume down. It slows the enzyme way, way down, effectively turning it off. That's brilliant. Why is that brilliant? Because by controlling this one enzyme, this product will be made in very tiny amounts, which means that this product is going to be made in very tiny amounts, et cetera, et cetera, et cetera. I control one enzyme, I control the whole pathway. Control one enzyme, I control the whole pathway. That's really powerful. Okay? What I've just described to you is a phenomenon we call in biochemistry feedback inhibition. Feedback inhibition. Okay? Feedback inhibition occurs when the end product of a pathway inhibits an enzyme at the beginning of the pathway. The molecule made at the end of a pathway inhibits an enzyme that catalyzes a reaction at the beginning of the pathway. So if CTP concentration starts getting high, it turns its own synthesis off. That's pretty cool. It's a very nice but simple balance. Too much CTP, pathway stops. Too little CTP, pathway starts. Yes? The amount of CTP will control whether the ATCase is active or inactive, or loud volume or low volume. All right. There's one thing that controls this enzyme. This enzyme is interesting because there's actually three things that control it. All right? Second thing that controls it is another nucleotide, ATP. Now, those of you who remember your nucleic acids, CTP is a pyrimidine. What is ATP? It's a purine. When we think about the structure of DNA, how are they paired? Purine to pyrimidine, right? If we have too much CTP, we have too many pyrimidines, right? It turns off its own synthesis. What if we have too much ATP? It means we don't have enough pyrimidines. ATP turns on the synthesis of pyrimidines. That's cool. We start to see the balance that cells can exhibit. We start to see that balance that cells can exhibit. That's really important. Okay? ATP turns it on, CTP turns it off. Right? I said there were three things that affect this enzyme. The first two bind to sites that are apart from the active site. All right? They're not anywhere near the active site. We'll see that next time. The third molecule actually is found at the active site. It's found at the active site. What do you suppose it is? To be found at the active site, there's a good exam question. To be found at the active site, what would it have to be? Have to be a substrate, right? What's the only substrate I've given a name to so far? Aspartate. Bingo. Okay, I always give you enough information to answer a question. You guys had enough information, you didn't realize it, right? Aspartate will activate the enzyme also. Aspartate activates the enzyme. All right, that's a good place to stop. I'll talk more about that next time. See you guys on Friday. And if you bring Dad, we have a surprise on Friday, by the way.